huge thank you to Liang and Arena for inviting us today to talk with you about this work. Like everything in mitochondrial medicine, it wasn't that straightforward, but it looks pretty easy looking back. I couldn't have done any of this without Elizabeth. Elizabeth really was the driving force behind this effort. And so I'm going to today just give you an introduction to what we tried to do. And then Elizabeth is going to take you into the nitty-gritty detail. So this is just some of my disclosures. I'm on the scientific advisory board, a research collaborator, consultant, or a PI of clinical trials with multiple partners in mitochondrial disease therapeutic development. Hopefully, it's a pleasure really to speak with this bioinformatics focus group. We created around 2012 a consortium known as the Mitochondrial Disease Sequence Data Resource, or MSeq-DR, and the website went live in 2014. I'll show you some analytics in a minute of who's using it, but it's been used and kind of created in a grassroots way by bioinformaticians and geneticists from around the world with the idea that our bioinformaticians, Dr. Zhao Gai and Li Shuangshen, they never used a pen, and yet every doctor, and I suspect Dr. Anselm as well, we always write notes on napkins and whatever we can to try and save information to figure it out later. So we really wanted to create a resource for geneticists and informaticians where all of the knowledge was centralized for the very many genes and variants and phenotypes, and really standardize the field, understand what variants cause disease and provide tools so that you could easily click to every other place on the internet where information might be needed that's useful to interpret a variant or a gene, or to better understand a phenotype, or to analyze your own data. So I'd highly encourage you to log in if you haven't. We host huge amounts of genomic data. I could speak to you, an hour just on this resource. We've had multiple publications. I refer you to the 2016 Human Mutation paper, which is the best bioinformatic description of the resource. And we now really link to tons of clinical data from laboratories that they've donated, lots of research data, and it's really been a lot of contributions from people all over the world to create the tools and the analyses. And I'll just point out in the upper corner, there's a mode for clinicians to make the resources available the way a clinician might think or prioritize the information. And if you click on that button, you can come to the bioinformatician mode, because there's some differences, as you might know, in what a bioinformatician might be looking for. So we try to make this useful to all people, and we're always looking for feedback and participation. Elizabeth coordinates a monthly call on the fourth Monday of each month at noon, where we give updates on the MSeq-DR consortium, on projects and partnerships, and looking for new ideas. So I'd encourage you to join us. This is an example of the Google Analytics from the last 5 years alone, showing we have about 250,000 page views, so around 50,000 a year. And you can see sometimes there's more than others. Very interestingly, the #1 page people come to, or the top 2, are really the phenotypic information. There's 200 plus phenotypes of mitochondrial disorders, and we've organized them all into an ontology using HPO terminology. You can also see there's a lot of other information, for genomic and analytic tools. So for both haplogroup and heteroplasmy and exome and genome analysis. And we're going to be creating in the future additional tools. We're partnering with the United Mitochondrial Disease Foundation, who's funded Dr. Shen, our bioinformatician, for the last 8 years or so, to now create a way for families to directly put their de-identified genomic data into this resource, so people can query it in an MSeq-DR OpenCGA instance to help solve the unsolved, and of course understand the finer points of mito disease. But this isn't really the focus for today, so I'll move on, and I'll just highlight that as an offshoot from the website, recognizing that that's a place for really informatic savvy people to go, we created a book, both an electronic version and a printed version, which summarizes, in 1 or 2 pages, every gene that causes mito disease based upon literature review. And I think 258 genes or so made it into the first edition as passing the criteria for causing any human mito disease. And there's about another 150 more that we're curating for the next edition. So I'd encourage you to keep this as a resource for research and clinical utilization to better understand how a gene can present across the life spectrum and the organ spectrum, what therapeutics might be available. And then there are links from each page on the electronic version that go back to the MCDR website, where you can find all the updated variant information. And we work very closely with ClinGen and ClinVar, and we have an accession number where we might have more information on a given variant that might be in ClinVar, and they link together. So this is just the background to say that there's still a lot of knowledge that's needed about mitochondrial DNA and how the variants in the 37 mito genes cause mito disease or don't. And I'm going to switch over to Elizabeth now. Elizabeth, do you want me to keep sharing, or do you want to share?
Yeah, I'm good. I can just keep on going.
Okay.
Yep. So thank you, Marni, for that introduction and the MCDR overview. And really how MCDR ties into what I'll be talking about is that was kind of where all of the experts were originally, and then we were able to expand upon that as we all came together to just try to standardize the way mitochondrial DNA variants are curated and assessed and ultimately interpreted when it comes to answering a question of, is this variant pathogenic or causative of a primary mitochondrial disease? So this is just a slide. I'm sure you've all seen variations of this before, but it's really just to remind you that we all actually have two pools of DNA. The nuclear DNA is definitely the more well-known and more famous one, but we do have a second pool of DNA, the mitochondrial DNA. And just two things to keep in mind as we talk about how we thought about standardizing the interpretation. Number one is in the upper right corner, you can see in the picture of the mitochondria, is we don't just have, we don't just have one mitochondria per cell, there's many mitochondria. And then within each mitochondria, you actually have many copies of mitochondrial DNA. And then given that, what can occur is there can be a mix in these copies of DNA, a mix of wild type and then a variant. And that mixture is called heteroplasmy. And that mix, or the percentage of mitochondrial DNA or mitochondrial genomes with a variant and those without, can actually be different among members of the same family, but also among tissues in the same individual. So you can have some tissues that have higher levels of heteroplasmy, some that have lower to actually undetectable levels. So always important to keep in mind. So again, thinking of why we had started on this project and really why it's important is because it's really important that we have consistent interpretation of mitochondrial DNA variants. Number one, for the families and the affected individuals to really end the long diagnostic odyssey that many of them go on. And furthermore, mitochondrial disease is kind of famous for being heterogeneous and nonspecific as far as symptoms go. We have showed that the patients have on average 16 clinical symptoms, and again, they're oftentimes nonspecific with a lot of overlap in other metabolic conditions. So it's important to be able to identify the etiology if it's there, but then also it can add a fairly significant burden to the clinicians as well as the families if there's inconsistencies in how diagnostic laboratories call the variants. Because again, thinking back to the concept that of heteroplasmy and having different levels of the variants and different copies of your mitochondrial genomes, what can happen is if one lab calls a variant pathogenic, and then we go and look in another tissue to get a sense of the mutation load in different parts of that in that individual, and that second maybe the diagnostic lab doesn't call the variant. Did they not call the variant because it's benign? Or did they not call the variant because it's truly absent in that tissue? And both those scenarios really have different outcomes as far as the interpreting that variant and having a better idea of whether it is associated with someone's medical concerns or not. And then, of course, diagnosis is very important for very many reasons. Currently, in the field of mitochondrial disease, there's many emerging and promising clinical trials. And for all of them, you need a confirmed genetic etiology. So it's really important for eligibility criteria for clinical trials. But then, of course, to provide appropriate medical management, and then family planning and recurrence risk. You know, the options are very different for individuals that have primary mitochondrial disease because of a nuclear DNA etiology versus a mitochondrial DNA etiology. So it's really important that we give an accurate diagnosis for that as well. So thinking a little bit about the scope of the work that we had done, we wanted to make sure it was very clear that our goal in trying to standardize this variant classification was really to assess the pathogenicity of these variants in the mitochondrial genome as they relate to primary mitochondrial disease. So that's a different question, whether there is some significance of variants in other diseases, such as cancer, or variants that might be acquired over time. And then we also know that there's been some correlations between certain mitochondrial DNA variants and traits or characteristics, like predisposition to common diseases or longevity, or protection against certain conditions. So that's really outside the scope of this work. We're trying to think, in this individual that has a primary mitochondrial disease, could this variant be pathogenic? And that is a separate question, though, of if a given variant is necessarily causing someone's disease, because in that case, you really need that clinical correlation with the physician treating the individual, because you need to take into account heteroplasmy and the threshold effect, meaning that when a variant reaches a certain level of heteroplasmy, it can cause disease manifestations. And that's actually different for almost every variant. And there's not one universal guidance that we were able to give for that. So I know that's not the most satisfying answer, and that question comes up often, but it's really just trying to answer, is this variant pathogenic? And then hopefully somewhere down the line, the issue of threshold effect and what level needs to be reached to be causing someone's disease will be addressed.
Yeah, Elizabeth, if I can just join in, that was spot on. I think that's a key point. When we're doing curation, we're curating published literature. We're not curating people walking in off the street, and so I think that's a confusing point for people, but essential to understand what we did. Thank you.
Yep. Yeah, so just a little bit on the background of this work. So it took very many years. I don't know if we totally knew how long it was going to take when we had started, but it was certainly a challenge bringing together people with background in the mitochondrial genome and mitochondrial disease, as well as the folks at ClinGen, or the Clinical Genome Resource, who to that point had been working on specifications that were primarily rooted in the nuclear genome, to try to bring those two groups together and have everyone agree with how those criteria for classification can be applied. So again, we started, I guess, close to 5 years ago now, and we had attained approval last summer. And throughout that time, we had really brought together the very many experts when it comes to the mitochondrial genome. So we were very diverse. There were clinical geneticists, genetic counselors, lab directors from academia, as well as commercial labs, individuals and experts with predominantly research focus and expertise, as well as bioinformaticians, who we know are really important to help us understand the databases and the tools that are out there to facilitate this classification. So now we'll just jump into how we had thought about kind of updating the existing guidelines, because that was really our task, was to go off of that really important Richards et al. Paper from 2015, where they had proposed variant classification standardization, again, for the nuclear genome. There was some mention of mitochondrial genome. I think it was a couple of paragraphs. So it was great that they had touched on it, but it really needed to be reviewed and then specified for the mitochondrial genome. So we kind of started at the top. We thought about what databases we need to consider for this work, what in silico predictors need to be considered, and then we kind of dove into each of those criteria for variant assessment. So thinking about databases that are most helpful, this is a table from that we had organized for the paper published last year. And we really tried to make clear, what databases are helpful when you want to look at allele frequency or our sequence repositories. What databases are helpful when it comes to variant pathogenicity assessment. And then what databases are helpful if you have a patient's mitochondrial DNA sequence that you want to analyze. So we started at the top with MitoMap. That's probably the one that most of you are familiar with. And we were very lucky that the individuals who did the curation for MitoMap, Marie Lott, Vincent Procaccio, and Doug Wallace, they were very closely involved in this effort. And then, of course, MCDR is the database Marni had told you about at the beginning. We have just some information listed here about the different tools and resources in those, as well as MitoSNP score. This was a database developed by a team in India. If you're all bioinformaticians, you might be able to work this database a little bit better than us that are more clinician focused, because you do somewhat need that background. And then lastly, HMTDP, which was developed by Marcella Adabinelli in Italy, but also has a resource of mitochondrial sequences to help ascertain allele frequencies, as well as predictors, and then tools for sequence analysis. And I will say this paper is already somewhat outdated because we don't have the NOMAD data set on here. And they actually do have data from mitochondrial DNA sequences. And we have, heard from their team and reviewed the data, and it is reliable what they have in there, and actually is going to be a good source for allele frequencies, variant frequencies in certain haplogroups. So we'll have to include that in the next iteration of this paper. So again, just to refresh all of your minds, you might be much more familiar with this Richards et al. Paper from 2015 than some of us, but really what we wanted to do, or really how this assessment is set up, is that they have criteria for pathogenicity and criteria for benign status. And then the different lines of criteria are really weighted, whether it's very strong, moderate, supporting for pathogenic, standalone, strong, and supporting for benign. And then when you combine these different lines of evidence, you get to that 5-tier classification system, whether it's pathogenic, likely pathogenic, VUS, likely benign, or benign. So what we had done is we had gone through every single one of these lines of evidence, or every single one of these criteria, and really thought critically among our group is, or whether, can this be applied for mitochondrial DNA? There's very many special features of mitochondrial DNA, which I'll talk about as we go through each of these rules. But we really tried to say, is this rule applicable to mitochondrial variants? If it's not, can it be specified or, minorly amended to keep in the spirit of the rule, but make it work for mitochondrial DNA variants? And then if it wasn't relevant for the mtDNA variants, to remove it altogether. And all of that we did not once, but actually 3 times, because there are different gene types in the mitochondrial DNA. So there's protein coding or mRNA genes, rRNA, and tRNAs, and these each have unique properties. So again, we went through each of these criteria 3 times to really see if they were relevant for each of these types of genes. So at the end of the day, just 2 genes were able to get off without any changes. One is PS1, and then one was PM4, although these are really only relevant for those protein coding genes. So we did not change them. And then several criteria were actually removed altogether just because they weren't relevant for the mitochondrial genome. PM1 talks about certain hotspots or clusters of variants. There's not really any of those in the mitochondrial DNA. We know that certain positions in the tRNAs can raise concern for pathogenicity, but it's nothing that's uniform that the group felt comfortable specifying here. And furthermore, that's captured in several of the prediction tools that we will get to in a little bit. PM3 has to do with variants being in cis or in trans, and of course that's not relevant for the mitochondrial genome. We all know it's maternally inherited. PP2 really wasn't able to be applied. We know that there's a lot of variability among haplogroups. The rules about reputable source, we were really encouraged by ClinGen to remove, and again, we agree. You need to really critically assess every variant. BP1 really wasn't relevant because we know truncating variants are not the predominant class of or pathogenic variants. And then BP3 we were able to remove just because there's not a ton of indels or repetitive regions, and they're actually known to be benign. So now digging into the variants that we specified or made some minor amendments to, the first rule is really about null variants, nonsense frame shift variants. So this was one that we thought a lot about, especially because this is very strong. It's the highest line of evidence for pathogenicity. So our group had decided that, even though, deletions can be somewhat tricky to assess, that those large, common deletions, at the very least where there's one gene deleted, they're always pathogenic. This genome is small, there's only 37 genes, and they're all critically important. So if one's deleted, that's always going to be pathogenic. And then thinking again about smaller deletions, nonsense frame shift variants, well, number one, those concepts are really only relevant for protein coding genes, but we had to think critically about these because in the first iteration of these guidelines for interpretation, these tend to be weighted higher just because we know in the nuclear genome there's nonsense mediated decay, or splicing can be affected. And for the mitochondrial genome, we actually don't have any evidence that nonsense mediated decay happens. And there is no splicing of the mitochondrial genome. So we were able to amend this flow chart, which was published by the ClinGen Sequence Variant Interpretation Group a couple years ago. We were able to just kind of take out the different parts that weren't relevant for the mitochondrial genome, and then amend what was there. You'll notice the initial guidelines did have guidance on initiation codons. That's something that our group really wasn't able to give guidance on just because they're so rare. There was no pilot set that we were able to test any proposed specifications on. So unfortunately, that's something that we weren't able to give guidance on. But those other variant types we were, as we have in this flowchart. And again, that is for protein coding genes. We know things are a little bit different for the tRNAs and the rRNAs because nonsense and frame shift really isn't relevant. These are not protein coding genes. And furthermore, small deletions and duplications in these genes are rare. So similar to the initiation codon problem, there wasn't a large training set that we could pilot any specifications on. So we really just encourage, if you were to see one of these in a patient or a variant that you're analyzing, to just look at the location of the variant and any possible structural effects that might come from a deletion or duplication in that tRNA or rRNA. So then we moved on to the criteria surrounding nucleotide position and amino acid position. And we didn't have to change too much here for the protein coding gene. So PS1 really centers around, you see a variant that causes the same amino acid change as a different nucleotide variant. And then PM5 centers around an amino acid change at the same position, but a different amino acid change than, again, a previously characterized pathogenic variant. So all that held true for those protein coding genes. What we did specify was that if you were to see a different nucleotide change at a same position in a tRNA or rRNA as a known pathogenic change, that could be a supporting line of evidence. And then we slightly amended BP7. So this one centered around synonymous variants at a conserved position. And we just amended this slightly. We took out the qualifier of conservation just because we had captured that in those in silico predictors, again, that I'll talk about in a bit. And the group felt comfortable leaving this at only synonymous variants because we know when you're looking at a nuclear DNA variant, you need to be somewhat cautious of synonymous variants with a concern that a cryptic splice site will be generated. But again, there's no splicing in the mitochondrial genome, so that's not something to be concerned about. So this can be included as a supporting line of evidence for benign. And then we had moved on to the rules looking at de novo occurrences. And again, this actually didn't take too much updating. The ClinGen SVI, Sequence Variant Interpretation Committee, they give a nice guidance where it's a table and it's kind of counting up de novo occurrences, giving them points, and then the points correlate with the different lines of evidence, whether it's very strong, moderate, or supporting. And they give kind of different levels of phenotype. And for the mitochondrial, for primary mitochondrial disease, that's, the phenotype is consistent with a gene or genome in this case, but not highly specific. But we did want to bring a few things to everyone's attention when you're considering if a variant is de novo or not. And that's, of course, centering around the sequencing technique. So we know that Sanger sequencing, the older techniques, can't really detect reliably heteroplasmy levels less than 30 to 50%. And that's really important because if you have a patient where you find a variant and the mother only undergoes Sanger sequencing, and it's not there, that variant might not be de novo, it just might be present at below that detection level of 30 to 50%. Whereas NextGen can really detect heteroplasmy levels as low as 1.5%. And the other thing to keep in mind is that it's really crucial to test as many tissues in mother as possible. As I said at the beginning, that sometimes some variants are only present in certain tissues. So if you have a patient where you find a variant in a skeletal muscle, but you only look in mother's buccal sample or only look in mother's blood, you might need to just test some additional tissues just to be as sure as you can be that that variant is truly de novo. And then thinking about functional studies, this was something our group had many meetings about, and we considered extensively. So really what we want to consider for functional validation as far as the mitochondrial variants go are hybrid studies and single fiber studies. So these are not something that's offered universally in a CLIA lab or anything like that. So at the current moment, so we were a little bit handcuffed where we had to downgrade it to supporting. Hopefully over time, there will kind of be more standard and universal parameters as far as these functional validation assays go. But currently they're only listed as supporting. So just to give a little background in case anyone's not familiar with hybrid studies, really what they center on is you take standard cell lines that are depleted of mitochondria and then merge them with your patient's mitochondria. So now you essentially have a consistent nuclear DNA background. And what you hope to get is if your patient is heteroplasmic for a variant, you hope to get clones that are homoplasmic for the wild type, and then compared to clones that have variable levels of the variant, whether it's high levels of heteroplasmy to homoplasmy for the variant. And in this scenario, you essentially have two cell lines or more cell lines where really the only difference is this mitochondrial DNA variant. So it's really a great test as well as far as functional validation goes. So when you think about hybrids, we did put forth some criteria that needed to be met to score them, even as supporting evidence. And that's first, that you have a biochemical deficiency in a patient cell line. So for example, if you're looking at a variant in a gene encoding a subunit of complex 1, your patient has complex 1 deficiency, you need to see that to then confirm if that was transferred to those cell lines. Because what you can do is actually look at oxidative phosphorylation, electron transport chain enzyme activities in the mutant versus control cell lines to see if you see any decrease in those corresponding respiratory chain complexes. So first, you need to see the deficiency in the cell line to then confirm it's transferred to the mutant hybrids. And to reach, what that deficiency might be, the group ultimately decided that it would be less than 20% of normal or more than 2 standard deviations below the control mean. And there's also been some studies that have been really nice where they actually showed that in hybrids, they might have a hybrid line with 60% heteroplasmy, 80% up to homoplasmic mutant. So for some of them, you can actually see that as the heteroplasmy level increases, the complex 1 activity, for example, decreases. So that's always strong evidence for pathogenicity of a variant. And then again, you need to see that there is a considerable mutant load in the variant. So at minimal 60%. And then of course, whenever possible, the studies need to be reproduced and consistent. And then thinking about single fiber studies, these have to do with muscle fibers from patients where you actually analyze single muscle fibers for oxphos activity and then heteroplasmy levels. So if you were to see a decrease, again, correlating with a high heteroplasmy level, that can be considered for supporting evidence of pathogenicity. So then moving on to variant frequency as well as haplogroup defining variants, this was also something that the group had thought a lot about and had many discussions. So it was somewhat tricky to define a cutoff for pathogenicity for mtDNA variants. When we were doing this project, I think there were at most 50,000 sequences in the population databases. So it was really challenging to, determine what cutoff would be a clue that this variant is pathogenic. Of course, if the variant's really novel or absent, that might increase your suspicion. But we were encouraged to put a number as best we could on what cutoff would be, on that cutoff would be. So we had done a lot of work. We looked at all of the variants that were confirmed to be disease causing in MitoMap. We looked at all the variants that we knew were haplogroup markers or haplogroup defining, and we had ultimately come to this less than 0.002% cutoff. There are certainly some benign variants that do meet that cutoff, but that was the kind of taking the conservative approach. And then when you think about benign variants and haplogroups are really just sets of single nucleotide variants that can define the maternal lineage. They're passed down over time. They kind of form these radiating patterns where you can track ancestral lineages. And those are by definition benign, but they can pose some challenges because if it's a novel occurrence in a different haplogroup, it's always not necessarily clear that it would be benign for that person. So when thinking about that, we really decided to specify that if the individual is a member of a haplogroup and you were to see a variant that is known to define that haplogroup, well, that's by definition benign. Or if the variant is present at greater than 1%. And then one step below that would be supporting evidence of benign status, and that would be a variant that's present at 0.5 to 0.99%. And again, we had talked about this in the paper. If anyone's interested, we had, again, just cataloged all the haplogroup defining variants, all of the presumably benign variants in MitoMap, and they had fit in with these cutoffs. And I have the websites listed here where Marie Lott and the team at MitoMap was actually able to catalog the most common haplogroup markers and haplogroup associated variants. So then switching gears a little bit, so thinking about segregation and presence in unrelated probands. So these are aspects that are certainly captured in that 2015 Richards paper, but really had taken some consideration for the mitochondrial genome, because we know that, again, given that we have many copies of mitochondrial DNA per cell and heteroplasmy levels can vary among, single individuals, they can also vary among members of the same family. So just saying a variant is present or absent isn't really enough to say whether it's segregating or not. You really need to correlate that individual's affected status. Do they have any of those non-specific symptoms that could be related to primary mitochondrial disease, as well as heteroplasmy levels? So what we had ultimately decided on was that a moderate line of evidence for pathogenicity would be if the heteroplasmy as well as disease manifestations are segregating in 5 or more maternal family members, and then 2 to 4 family members would be a supporting line of evidence. And then on the flip side, for benign evidence for a benign classification, if you were to see higher heteroplasmy levels, number 1, in a healthy unrelated adult or a healthy family member looking in the same tissue, that would be a strong line of evidence. And then supporting, again, would be similarly just if a different tissue was tested than the individual or proband. And then, of course, finally, support or a strong line of evidence would be if disease is segregating in paternal family members. That should always be a red flag that maybe the mtDNA variant is not the cause in that family, or a lack of segregation. So those are the rules surrounding segregation. Then when you think of the rules surrounding whether they're case control studies or the presence of variants in unrelated affected individuals compared to control. So we know for primary mitochondrial diseases, these are individually rare, so there's not going to be those large case control studies. But you can look and see, as Marni was saying, in literature, or if you have a diagnostic lab and other probands that you had tested, to see if the variant does seem to be enriched in affected individuals compared to healthy individuals. So what we had ultimately ended up deciding was that a strong line of evidence would be if there were 16 or more individuals that had this variant, 4 to 15 would be moderate, 2 to 3 with the stipulation that they are from different haplogroups for supporting. And when we think about proband, this was also something that took a lot of consideration from our group, because again, we know that mitochondrial disease is notoriously nonspecific and there's no one great biomarker. So to consider someone affected, we would say that that individual had to have one of those classic mitochondrial disease clinical syndromes. Those are the MELAS, NARP, MRF, MID, very many acronyms that are out there. In the absence of that, there's was a paper, several papers in 2007 and 2008, where they really listed red flag features and the non-specific features of mitochondrial disease, suggestive lab abnormalities. So we have up here the kind of combination of those red flag features, non-specific features, and lab abnormalities that you can ultimately say, this proband does meet criteria for primary mitochondrial disease. I can count that individual in this line of evidence. And then moving along, so we know that in silico predictors can be helpful. Of course, we always take them with a grain of salt, as you do for nuclear DNA. The problem is that a lot of those the predictors commonly used for nuclear DNA, SIFT, polyphen, those have been shown to not be good for mitochondrial DNA variants, so we didn't recommend using them. But there were several predictors that did hold up well. One was APOG. This is really a meta predictor that takes in a couple of different predictors, conservation, location of the variant, so on and so forth. And gives you a nice score where greater than 0.5 is evidence for pathogenicity, less than or equal to 0.5, they consider it neutral. And this is great for the protein coding genes. This is actually only for protein coding genes, these as apogee predictor. When you think about tRNA variants, there's different predictors that are out there. And there's actually two that are most helpful. One is mitotyp, and then another is HMT-VAR. HMT-VAR actually does have predictors for the protein coding genes as well, but that's actually included in the apogee predictor. So going back to the tRNAs, we just have listed up here kind of if, both of these predictors are predicting pathogenicity, you can apply that supporting line of evidence, that PP3. If they are conflicting in whether they're saying the variant is deleterious or benign, you really can't apply this line of evidence. And then if they agree that it's a neutral or benign, no effect, it can be evidence for benign. You'll notice here our RNAs, unfortunately, there wasn't a predictor out there that was easy to use or intuitive and we were able to use for this guidance. So maybe in the future, something will be put forth. But right now, again, as far as the computational evidence goes, really, we just have listed here tools for protein coding genes and the tRNAs. So then moving along for phenotype, this is a supporting line of evidence, and kind of the spirit of this rule is that the individual that has the variant has a phenotype consistent with the condition. So again, we use this a little bit when you're, counting probands, but for this, we had specified it to capture respiratory chain enzyme activity. So we know that this is really still a test that's commonly used to help come to the diagnosis of primary mitochondrial disease to kind of gather evidence for or against that being the cause in someone. So if you were to see a decrease in complex 1, for example, in a patient's muscle or liver, that would be something that you can say, okay, this patient has complex 1 deficiency, they have a variant in this gene coding a subunit complex 1. This is evidence for pathogenic status of this variant. We specified if you were to look in a fibroblast cell line, we know some patients don't want to do muscle biopsy no matter what. So if you were to look in a fibroblast cell line, it would just help to look in, multiple unrelated probands or different individuals that have this same variant, to see if a deficiency is seen. And a deficiency is really defined by the clinical lab that's doing it. So the labs that do this, again, CLIA labs here in the States, there's other certificate programs, around the world, but really whatever that lab recognizes as being deficient, we could use that in this category. And again, typically, the criteria is class B been less than 20% of the control mean. The one caveat here, and this can certainly pose some challenges, is that we know that, again, if we use the complex 1 example, there's very many subunits of complex 1, very many assembly factors. We need products from both genomes for these respiratory chain complexes to function properly. So if you have complex 1 deficiency, and for example, you only sequence the mitochondrial DNA, well, how do you know there's not a nuclear DNA etiology that's causing that? So in order to apply this, you really need to exclude those other etiologies to the kind of best of current abilities, typically by exome, genome, and mitochondrial DNA sequencing. So just wrapping up here, this was the 2 rules that kind of center around alternate disease cause. We certainly apply these carefully. So how we have them written is that if you have a variant in a patient and that patient has a separate mitochondrial DNA variant that's pathogenic, well, the variant in question may not be causing that individual's disease. However, we know that there are case reports of individuals with 2 pathogenic mtDNA variants. So you certainly have to apply this with caution. And then thinking about nuclear DNA, if you have a mitochondrial DNA variant, but you have a patient with a nuclear, DNA related disease, again, you might think that mitochondrial DNA variant is then likely benign, but of course we do know patients that have 2 and 3 diseases, so across both genomes. So just something to just be very cautious in applying. So that is it as far as the specifications. And I know we're getting to the end, so I'll just wrap up with a few examples. So the first case, this was a girl that was found to have the 10158T to C variant, an ND3, I believe it is. And she certainly had features consistent with Lee syndrome and mitochondrial disease. So if you are thinking about how you would apply these rules, you can see here there's multiple for this area, multiple reports in the literature of de novo occurrence. There's multiple people in the literature with classic features of mitochondrial disease. Alternatively, there's multiple families reported where the level of heteroplasmy is segregating with disease manifestations. It's predicted to be pathogenic by apogee. It is absent in those population databases, and there are really strong hybrid studies for this variant. So this would end up being a pathogenic variant. Moving on to case 2, this was also a girl with some features of primary mitochondrial disease. And she was found to have this variant 4735C to G. This is a variant that there is just a lot less information on. There's really no other cases in the literature, no hybrid studies. So you're really just kind of limited to looking at the in silico predictors and allele frequency. So you see this is just going to kind of be stuck at that variant of uncertain significance. And then lastly, again, a man with some features concerning for primary mitochondrial disease was found to have this variant at position 7476. However, you can see when you look at this variant, is actually associated with several haplogroups. And those that are haplogroup J2A, it's present in 99%. And it does have a fairly high allele frequency overall. Furthermore, it's predicted to be benign. So this would reach that benign classification. And then just wrapping up with some resources, of course, MitoMap, MCDR. Again, we are the ClinGen mitochondrial disease expert panel. So that's a link to the ClinGen website and our expert panel page there. And then just to finish here for future directions, again, we can definitely update the allele frequency as more healthy mtDNA sequences from healthy individuals are published and able to be queried, hoping to give more criteria for those hybrid studies, because there definitely are some variations, and then continue variant creation. There's very many variants that still need to be reviewed by this expert panel. So I just have my email up there if anyone has any questions, and then we can just wrap up here with those acknowledgements. So that is all we had. Happy to take any questions.
Well, thank you so much, Elizabeth and Marni. That was really very, very interesting. And thank you for your wonderful work. For us clinicians, it is so important. We so frequently just confronted with these questions, whether the variant that's reported is pathogenic or not, and we have to rely on interpretations of commercial labs. So my question is, how do you think this work will affect how these labs, like let's say GeneDx or other ones, how they review and report these variants, and whether this will be any change based on what you guys done.
Yeah, I mean, I think we certainly hope it'll be more consistent across labs. I don't know if you've, noticed that, some labs call every variant, some labs say normal, nothing, right? So you're kind of just hoping to find that middle ground where there's just consistency. You know, some variants are going to be just in the US until there's more cases published, more information out there, more population, healthy population sequences. But I think we're just really hoping to at least come to a consensus, you know, on what is the classification of this variant.
Yeah, and if I could add, you know, the people that were working with us were from, really, it was the clinical lab directors arena who wanted to better do this, do this better. They were facing this, you know, and seeing this as a major concern that they weren't sure how to interpret it. So we've had a lot of volunteer curators from Illumina, from GeneDx, expert panel members from a range of other clinical labs, academic labs. I think Christine Stanley was at 2 or 3 different labs over the course of this project. So I think it's really raised the rigor. And that's one of the real reasons we did this, that the laboratory directors would have guidance that would, you know, better help them. And I think the other thing is to just raise the criteria among or the rigor among our clinical community, that there is a way, you know, to think about these variants systematically. I know in our clinic, Elizabeth will go through all of this information. We probably need to create an easy tool in MSeqDR at some point so that clinicians can do this too. And we do have another funding proposal pending to do what we did for Lee syndrome, where we were doing gene and variant curation for genes and variants in both genomes that cause Lee syndrome, to now try to do it for all primary mito disease. So the hardest part was coming up with these rules. We actually had to merge, I don't know if you caught that, but we had to merge the ClinGen committee. There was an MSeqDR committee, and there was a clinical diagnostic lab ACMG committee, all trying to do the same thing around 2016 or 2017. And we had to agree to work together so that we had a consensus set of criteria that we could all agree. So that's one of the main goals of MSeqDR, is to create community consensus, you know, even as things like what is the reference genome, what is the right format for mtDNA genome. You know, we came up with a standard VCF file for mtDNA calls. What is the haplogroup tool. So I think your point is well taken, that we probably need to create an easier way for other people to access the ClinGen process. But we're happy to train anybody who would like. And as the mito disease expert panel, if there's people on this call that want to curate other genes or other variants for different conditions, you know, besides what we've done, we're happy to train you. And the rules for ClinGen are that you need to have 3 experts who did not discover the variant or gene from 3 different institutions. So now that we've created the process and the what's the right word, protocol, I guess, the guidelines, anybody can do it. So I do hope it helps us better assess these variants relative to disease. And I think the other thing that we really realized as we went through this, we all have to keep publishing. You have to publish your cases, because the only way we learn is by sharing. And a ton of information, people would have had cases that they never published. So you can only count things that are in the literature. So it's just a great reminder why people need to keep publishing their known genes or their variants every so often, or to submit it themselves to ClinVar, so that we can learn from it together. And of course, keep doing functional validation, very important.
Yeah, I agree with you. And, you know, as clinicians, sometimes we don't have time for publishing, but I was glad, like Ren Kubai from GeneDx kept bugging me about publishing data on MPFMT gene mutation. But also Elizabeth mentioned that we have to try to test mothers if they are negative, like on different other tissues, not just on the blood, but maybe on saliva and maybe on urine too, right? I mean, I tried to find a commercial lab which would do mtDNA sequencing.
Yeah, we can update you. We could update you on that. So that's clinically, that's always been a challenge. You know, getting insurance to cover a healthy person for mtDNA sequencing can be just an enormous battle, enormous amount of time and effort. So we actually developed a test here at CHOP that's open to anybody, that it's an mtDNA genome by NextGen. It has 3 long-range PCR products, and it automatically reads 2. And if something doesn't sit down because of the, you know, a primer issue, then there's a 3rd primer they can go to. So it detects heteroplasmy, and it detects deletions down to 1%, which is pretty neat. But it also, for deletions, it does that on a digital droplet PCR reflex. So if it's seen it, you not only know the breakpoints, you know the heteroplasmy level, because you can't rely on deletion heteroplasmy by NextGen. So we developed it's included in the test. But the other thing that I think is a major win for the clinical world is that every time you order it, they'll run one maternal sample for free at the same time. So you don't have to fight insurance. They accept the proband sample and the maternal sample. And like it could be muscle from the kid and urine from the parent. It doesn't have to be the same tissue as long as you send it within the week. So for us, that was huge. Plus we could also, if you needed it, can get a rapid result in 2 weeks, which you can't get otherwhere. So we had to just create the test we needed so we can get it. And then they've also now optimized. Elizabeth, do you have the full list of samples we can get? Urine was a big battle for people because of the degradation. So Baylor had stopped offering it. I think GeneDx now offers it and CHOP now offers it. I think CHOP also offers saliva. I think one or both offers buccal. But yeah, these are really important issues, and they're even more important when you want to get into the prenatal. And should we be doing, prenatal testing? Well, it depends. Like, is the mother a carrier or isn't she?
Well, prenatal testing is really tricky, right? It's just
that's a whole nother topic. We're getting there. So we now have ways for people to stay in America and send their embryo biopsy to England and then get the result back and implant it. So the problem there has always been the reliability of the testing and whether there's any US CLIA labs that will do it. And the long and short is no, but where you have the Newcastle lab will do it. And so we've just over the past year, and I don't know, Elizabeth, like 100 conversations have it going. So if you have families that need it, I think it's now possible. Before then, people actually had to travel, for PGD there and implant. It was very, very hard on people. And the costs are very high.
No, it's a different topic, but I must say that like 15 years ago, I was able to do prenatal testing on a fetus already on CVS culture for 8993 mutation because mother was negative there, the child had Lee disease, and it was done by Baylor. And then they stopped doing, like everyone stopped in the United States.
So much of it, I think, is liability. So much of it is the testing. A lot of it was before NextGen. But I think there's a lot of European data suggesting that it's definitely worthwhile. There's definitely some reasons why it might not work, fertility related and heteroplasmy levels being very high and not finding one low enough. But I think that the truth is that it should now at least be considered and that it's more reliable, actually. The groups in Europe have shown it's more reliable to do the PGD 3-day biopsy on the embryo than it is to do CVS or amnio. So if people really want to know, it's definitely something I think we need to try to make more available for our families in America.
Right. And especially since hopefully we will be moving towards mitochondrial DNA transplantation or transfer.
Yeah, it's a whole nother topic, exactly.
Yeah. All right, I think we ran out of time, but thank you so much again. Elizabeth, maybe you can share a link with us to these seminars that you give every fourth Monday so that we can go to them. That would be awesome.
Definitely.
And if anybody wants to join our curation effort too, just let Elizabeth know. We are up to, I think, 50 experts from, I don't know, what is it, 11 countries on 5 continents, something like that.
Yeah. We're up to, I think, every continent now except Antarctica. So very global.
So we're always welcoming experts. It's not that much time and effort for experts. So there's curators. We're always willing to take more of curators to actually go through it. It can take hours, to curate one variant. So it's a labor of love. But I think the expert panels, once or twice a month, we meet and all the data is presented and the experts have to vote or ask more questions. So I think that's a very satisfying thing. If people want to participate, we definitely welcome any help people want to share at their time.